Okay. Who would like to volunteer to go first for the hack pitches? Come on down. Yeah.
Don't have to be like polished.
Our expectation is that these are not polished. Yeah.
Okay. So I had a much more refined idea, but I kind of wanted to take a more exploratory route since we have like 4 days. And I didn't know if like other people would be interested in this, but something that I'm really interested in is whole brain dynamical models of attractor landscapes and functional brain activity. And so I'm hoping to do some exploratory research using the HCP young adult data set, just because that was the one that was the most easily available. Initially, I was going to try to train on different disease data sets and then use them, but that was a little chaotic. And for like a general background, it's for people who don't know what those are. It's basically just that brain activity is dynamic, not static. And neuroactivity tends to settle into stable patterns called attractors, and the collection of possible attractors is called an attractor landscape. And so individual differences in these landscapes may relate to like behavior, cognition, and disease. And these whole brain dynamical models can kind of estimate an individual's attractor landscape from neuroimaging data, and that enables pretty interesting quantification of brain dynamics and instability. Um, and just as far as logistics, I mean, it's basically the normal software packages, the Nyler, NiBabble. I'm hoping to use Pandas. Um, but there would be like one more for hidden Markov models, so like GLHMM. So it would be pretty simple, and then just another layer of like HMMs. Yeah. And I have like a little timeline, but I feel like that's probably too much.
That's a great thing to talk about in your first meeting. All right, thank you. And thank you for going first, Monica. So now your job now is to put an entry in that spreadsheet and to post something in Hackstorming that just says, here's one of the pitches, find it in the spreadsheet. Okay, excellent. Who wants to go next? Yeah, come on up. Your speak up.
Hi everyone. So the idea that I have, so for my PhD, what I'm doing is developing these computational tools to understand how humans process complex naturalistic audiovisual stimuli such as movies. So I kind of wanted to continue onto that exploration and explore new data set that I thought so far the data sets, the movie data sets I found to be the most annotated one. So basically, this is this data set called Study Forest, which is based on people watching the movie Forrest Gump. So they have this data. They actually have the audiovisual movie data on fMRI data. They also have people just listen to the audio and have the fMRI data for it. But what I found the coolest thing about this data set was they had these annotations, very detailed annotations about the emotions and different kinds of intentions, such as like lies, irony, sarcasm. They also have very detailed data about annotations about like different kinds of physical interactions. So I thought that this was very rich, and I haven't worked with this data set before, so I thought it would be pretty neat to at least try to understand to what extent maybe social or like this kind of intentions that are present in movies change across time, because I think that's the kind of question I'm interested in. Is just like naturalistic, uncontrolled setting, a lot of information content just vary over time, and how do our brain process that? Now, obviously, fMRI data might not be the best to get to the very root dynamics of this, but you can still get a pretty good sense of a lot of interesting things that I think are still relatively underexplored. So as far as how it's going to look like, so they have like there's lots of annotation data sets. So what I thought could be, it could be neatly divided amongst the group members, who could like maybe, each of us could like pull look at one one of the data set, try to figure out how to set that up on the Jupyter Hub. If we have to, I mean, ultimately, I don't think that we will be able to get to any interesting scientific questions in like 3 days of coding. But I was just thinking that we could at least try feeding some encoding models and see these relevant regressors that we have access to, like emotions or like intents, like to what extent which regions are being predicted by them, and how that pattern change, and if we can even like replicate some of what the literature already know about where emotion is processed. I think ultimately what would be really cool is to see, okay, like, a lot of these things like emotion or like theory of mind, attempting to understand one's intention, these are very multimodal construct by their nature. And if we could tease apart to what extent the audio and the visual information is, like helping us process that construct, that would be very neat, but I don't think we can get there. I think, but since they have this bunch of data sets that each of us could kind of look at and like try to set up, I thought that this could be, this could be kind of divided, and we could kind of try to do some divide and conquer and try to see if we can predict, at least like recapitulate some of the findings the literature has, which we could just probably do with some encoding model, and maybe it could be achievable in the course of 3 or 4 days. So, yeah.
Okay, who wants to go next? Yeah.
Hello. All right, so here's a half-baked idea. I have been thinking about building a Python package or a tool to generate anatomical and functional labels for neurosynth parcellations, and this could be extended to be parcellation agnostic. And I was motivated to think about this because often in neuroimaging studies, we ask the question of which brain region is associated with a particular cognitive process of interest that we might be studying. For example, which brain regions are responsible for mental state representation. And to do such analyses, we often rely on functional parcellations, which divide the brain into parcels with similar functional properties. And neurosynth is a popular choice of interest, and that's something I worked with for the first time. And it offers 3 parcellations with 50, 100, and 200 parcels. And the higher resolution parcellations are powerful. They give us more spatial specificity, but they are also unlabeled. And for new trainees like myself, it has been very challenging to look at a brain map and tell what the heck is going on and interpret what parcel 0 or parcel 200 means. And a package that could take in a brain map or a parcel number and return a tentative anatomical location or its functional purpose could solve a real practical problem and could be just used in your analytical workflow directly. And I have some ideas about how this can be done. For example, cross-referencing a parcel's coordinates with another already labeled parcellation or an atlas. And I recently discovered Nightmare, which is a new package for neurosynth, which can help us get the cognitive processes strongly associated with a given parcel. Those are all my thoughts so far, but if you're interested in this, I would be excited to hear ideas. Thank you.
Great, thank you. We love half-baked ideas. Who wants to go next? Yeah, come on down.
Hey, hi everyone, I'm Tian. So for those of us who study fMRI,
Just
okay. So for those of us who study fMRI, how many of you have used Andy Springbok as a resource? Yeah, Andy Springbok, quite a few. Great. So I work at the University of Michigan, where Andy is too. So whenever I have questions about fMRI data pre-processing, he's one of the first people I go to. And at first I saw, oh, Andy, you know everything. And then I realized it's not how it works, because even in the Andy Springbok, he doesn't have too many resources on quality assurance, aka QA, which is one of the kind of least standardized parts in fMRI data processing. So right now, most of us learn quality assurance from senior lab members or comparing our notes with our peers. There are not too much standardized resources showing what different artifacts look like or how to decide whether a scan would pass the QA or fail. So what I want to do is to kind of build an open and community-driven GitHub page website with annotated examples, artifact QA, practical QA guidelines, and maybe even interactive quizzes on whether a brain will pass or fail, whether a scan will pass or fail. So if you ever struggle with QA like I did, or if you're interested in QA, I would love to have you guys. And if you're not interested in this project, but you have like bad brains, bad scans, or good scans that you want us to include as an example, I will appreciate your contribution as well. So the goal isn't just to build a website, it's to make neuroimaging quality assessment more reproducible and easier to learn. So obviously I cannot go to Andy with my 300 participants and ask him whether this brain will pass or fail. So I think it's really helpful to have some learning resources for the community. So we're gonna start with fMRI because that's what we know. At least that's what I know the most. But we love to think how these ideas could eventually extend to other neuroimaging modalities. Thank you.
I love the idea of community sourcing bad scans as a way to teach people, because those never make it out of the lab usually. Who'd like to go next? Yeah, come on up.
My name is Tongla, and I have the idea to explore the benefit of precision fMRI for multivariate analyses. So I actually developed this idea during Katerina Gretzen's talk. She talked about how networks are organized very differently across people, with lateral prefrontal cortex being the second largest variant network. And the number one largest network is the default mode network. And in my personal work, I usually do memory-related research using DMN, default mode network, regions of interest to do multivariate analyses. And from that talk, I kind of realized maybe we're doing something wrong. Maybe we are setting up an RI that's honestly sitting on the borders of different networks. So thanks to Katerina Gratzen and her amazing data set, she had this individualized network definition. And I wanted to see applying that network definition in comparison to a group or template network, could that yield in for say, significantly better multivariate analysis results? For example, brain decoding. And they have not only rest and state functional connectivity data, but also 13 different tasks, among which one of my favorite is the episodic projection, but I'm honestly so welcome for people to try out different tasks. And so some of the keywords of the work I want to do is task connectivity, rest and state connectivity, network definition, and also episodic projection. Yeah, thank you.
Great, thank you. Who wants to go next? Yeah. Good slide. Okay, so can I have you log into Zoom and just share your screen? Because if you just connect with nobody online, you'll be able to see it.
I can also participate.
Yeah, if that, yeah, that or if you want to go back, if you want to wait, it's fine. Yeah, okay. If you wouldn't mind standing behind, I'll just see. No, sorry, behind the podium.
Behind?
Yeah, so that lets people online see it.
Hi everyone. So my idea is to leverage the pre-trained knowledge of large language models to create a general purpose inference machine that can ingest any type of multimodal brain data with any possible clinical phenotype. So the idea is that there's a limitation of conventional machine learning algorithms in which we typically only train it to predict a single disease or single dimension. So the idea is that large language models have been learned to predict the next token probability distribution in a way that captures actually a lot of modalities connected to a lot of clinical phenotypes. So for my internship, I built a tool. It's called the Compass Engine, Clinical Ontology-Driven Multimodal Agentic Predictive Support System, that has been proven to be significantly predictive of 5 specific disorders without any pre-training, without any training of an actual model. So I'd like to explore how we can use this pre-trained knowledge of the models, of large language models, I mean, with open neural data sets. So I just want to further validate it. I've already used the UK Biobank, which is the largest Western biomedical database. But it would be nice to now use open source data sets so that I can actually use public APIs, which would give me more access to better large language models. So that's the idea. I can also share the paper or the internship report in the channel. Thank you.
Amazing. So yeah, if sorry, I should have said this earlier. If you want to share slides for this pitch, please join the Zoom channel and share your screen and make sure you're muted before you come down. But otherwise, you can put the slides in the channel and people can find them. Okay, who'd like to go next? Yeah, come on down.
Had to like circumnavigate the whole auditorium. Hi, my name's Heather, and my project that I'm pitching is very selfishly motivated. I have a chunk of my own functional brain data. It's me looking at pictures of different people in my life, so different kinds of social relationships, and I haven't touched it at all. And so I was thinking it would be really interesting to do some kind of like multiverse analysis with it. So like the method that my lab would probably use is like representational similarity analysis. So make an RDM of like this person's my friend, this person's my ex, etc., and then see how representations map onto that. But I think there'd be a lot of like different kinds of potential like things that we could do with the data. And also like I want to know what's going on in my brain. And I'm like happy to answer any psychological surveys you want me to answer and do a fun thing.
Okay. Anyone online who's I'm missing maybe? Okay, so there's no one jumping up online, anyone in the room? Yeah, come on down.
Well, I have 2 ideas. One is a little bit simple and the other one is half baked. So hi, I'm Manuel. The simple idea is mostly similar to what we did on the previous week. So we had this very large evidence about the needed white matter connectivity to actually perform the task of recognizing faces, but it's mostly from patient data, so lesion data. So we have been trying to set up this reproduce that same idea of which white matter tracks do you need to actually perform better at recognizing faces, but with healthy adults. So we could use the HCP data, so we could rerun the diffusion data from the HCP and correlate it with this task that participants had to do to memorize faces and then recognize them afterwards. It's an N0, N2 back, and then we have data after they left out the scanner. So we try to see if we can reproduce what the tracks that are needed to recognize faces better, because we kind of started from the idea that face recognition is a skill that can vary along a spectrum. So like maybe people who are face blindness from birth, they just are lower in the spectrum. So we could use kind of that same idea for the development of prosopagnosia, what that is called. So that's one idea. Do it everything in Python, kind of learning to do the diffusion and apply it on the HCP data. And the half-baked idea is that we have been trying to model those same lesions on brain data, but for agnostic patients who could not recognize faces or other objects, but do it with neural networks. Can we test some hypotheses about the mechanisms behind perception when we model them in neural networks? And it's a bit difficult to actually do that because neural networks don't easily get damaged so easily, or there are several ways of doing that. And I just found out this last week that there's one network that came out, the V1net, that tries to reproduce more neural network that is more biological realistic. So we could try to set up this network or a similar network like HMAX in PyTorch and try to run lesions onto the network and see if that data comes out similar to what patients cannot do when they have lesions that produce agnosia or perception problems. So that's the half-baked idea. Well, thank you.
Who's next? And that's gonna pitch project? Yeah, no.
Okay. I'm sorry. Okay, so I'm Emily. This definitely isn't like novel. I'm more just am thinking about like what skills I want to practice and learn and what data is readily available. So I was thinking of looking at, I saw the young adult HCP has some task data, and I want to practice doing some machine learning stuff with fMRI specifically, either diffusion or functional connectivity, because I haven't done those before. So trying to use the brain to predict task performance on a delay discounting task that they have in that data set, which a lot of people have done before. So this is not new. But if anyone else wants to work on this, that would be cool. Okay, thank you.
Just to be clear, I maybe didn't say this up front, but the goal of these projects is not for you guys to like publish something at the end of the week. It is for you to learn something, and we're here to help. And so I think projects that are designed for you to learn something are great. Okay, you have one? Yeah, come on.
Okay, hello everyone. This is gonna be very informal, but I'd like to try and find a like neurobehavioral predictors of maybe weight gain or weight loss in the NKI Rockland sample. So I kind of just really 5 minutes ago settled that idea. So yeah, very informal. We'll try to do what we can, but yeah, that would also serve me back home at my lab. So yeah, if anyone wants to work on that, that'd be great.
Who else? Yeah, come on down.
Okay, hi everyone. My name is Joe. I have a couple of half-baked ideas as well. And they're a little different and they're not necessarily neuroimaging related. So the first is to create a personalized best practices manual. I know we've a lot of talks about reproducibility and modularity. And personally, when I look at my analysis scripts, I know I should not be doing that, but I just like keep coding along. So I was just thinking we could like review past projects we've done and kind of just look at some things we can improve to bring forward into future projects, and that could be personalized to whatever you're doing. So we'd love to work with someone on that. The second idea is if you're interested in art or like poetry, I felt like it'd be fun to just like riff on some of the themes with learn from this week and just, I don't know, play with that. I don't know, just an idea. And then my last idea is if you're interested in neurosignatures, I would love to work with someone on that because I was really inspired by Wani's talk. And I'm personally very interested in attachment, and I was thinking we could, you don't have to be interested in attachment, but if you're interested in like creating a study design that would help you develop a neurosignature of your construct of interest, I think it would be fun to work together. Yeah, all right, thank you.
Great. We've actually never had anyone take us up on doing poetry as a project before, but we do stand behind that is a valid project. Okay. Anyone else got an idea? This is your chance to pitch to everybody. It doesn't have to be a full. Anyone online? Yeah. Come on.
Hello everyone. I would like to suggest the project that I've worked with. Like it's basically a retrotoping mapping, which tries to understand which locations in the visual field our visual area respond to. So, for example, our visual area has some coordinates, and so when you look at some scene, there is some, we can represent it as a coordinate, and we can say like specific voxels in this visual area respond to specific degree in this coordinate. So that is what is called retrotoping maps, and it takes quite a lot of effort to get those kind of data set. We need like hours of additional sessions to get those locations. So there has been some trials to automate this process. For example, using some templates or using some structured MRI data to predict individual differences of retrotoping maps. However, the biggest challenges for now is that the templates or models used for this generalization it depends on Human Connectome Project dataset, 7 Tesla. So it does barely generalize to any other data set. So the one I would like to do here is to curate multiple data sets that is available on multiple sites and do some QA on that and make some benchmarks on that. And after that, another thing I would like to do is to train some deep learning models that depends on self-supervised learning to train the models with only structured MRI data and tries to generalize the pre-trained models to this specific retrofit mapping prediction task. Yes, thank you, everyone.
Hey, who else? All right, going once, going twice. Okay, so okay, I figured someone did jump up as soon as I started to find out.
I haven't joined with a couple of other people. I've heard nice things, but maybe I'll just throw it out in the open.
Yeah, go for it.
Sorry. Good that you asked them.
What's that?
The good that you do.
Oh.
So basically, this is me taking a fall for you guys to take some more time and pitch more half-baked ideas. 1 second, where was I? Yeah. So good morning, everyone. Here in Zoom. How many of you guys are familiar with metabolites, MRS stuff? Any people? Okay. couple of hands. All right. So in this niche place where metabolites kind of measure the neurotransmitters, signals and the strength, it would be cool to know how that relates to connectivity and if there are individual differences with it. So HCP interestingly includes a 7 Tesla metabolite release for older adults, HCP agent. So I would like to leverage that data set to see how the strength of metabolites will start with something well known like GABA and glutamate, then proceed to creatinine and other lesser known and more individual differences which could create, and then see if they map onto functional networks like it's known, I think GABA has signatures in PCC, so maybe the posterior deformer network would be involved, but to what extent and the strength is kind of unknown. So anyone want to come with me on this exploratory journey, then I would be happy. There's another half-baked idea. So over the time, I've been interested in looking more at the dynamic causal modeling, which is a little different from someone mentioned dynamic networks. Basically, dynamic causal modeling looks at effective connectivity. So I want to do that in EEG because of the time is really a thing which fMRI doesn't do very well. Sorry. So, but EEG tends to do that very well. So there are couple of large scale open set data sets which have auditory oddball and visual oddball tasks. Nothing new hasn't been known. We're just testing older models, but in a larger set to establish that the same pathways include, for example, if it's auditory oddball task, does the signal travel from primary auditory cortex, A1 to the posterior belt to STG to the motor cortex and back, and something simple, but yeah, testable on a larger scale. So yeah, that's the second idea. So yeah, that's will be my 5 minutes. Sorry. Thank you.
Please talk about it. All right, anyone else? Okay. Great. So I'll just say a few more things here. We'll talk more about what we expect for the final presentations later in the week. For now, don't worry about those where they're, well, yeah, just don't worry about them yet. We'll talk about that probably on Wednesday. I would just encourage you all to, like I said, go find a place to hang out and be prepared to spend the first part of today talking to people who are maybe interested in the project and want to know a little more about what we might be trying to do. If you think you're going to need data on the hub or you're going to need data that's not easy to access over like Cloud Pathweb, please let me and Ariel know. We can work with you to try to get the data there. It is not possible for us to put every data set on the hub. I'll just say some data sets have restricted data or you need to register or something like that. We can help you navigate that. Of course, if you're using a data set that you can just access freely, that's great. By all means, do that. There was something else I was going to say, but I forget. I will, if I remember it, I'll put it in the announcements channel. Great. I think at this point we can say, go ahead and start hacking and